So, hi, my name is Jillian Belluck. I am an undergraduate student at Brown University. And this summer I was working in Jim Giovannoni's lab, and I was working on studying the phenotypic variation in tomatoes.
And now, can you introduce some of the methods, some of the results that you got from this, and what are some of the key takeaways that you've found with this project?
Yeah. So the purpose of this project is to use computer vision to phenotype a lot of different wild and cultivated tomatoes. So I can show you here. These are actually some of the tomatoes that we're working with. So this species of tomato is called Solanum cheesemanii. It's one of two species that is native to the Galápagos Islands in Ecuador. And these tomatoes, as you can see, they're very small. They're about the size of blueberries, but this is as big as they get. But there's a lot of phenotypic variation in size and also in shape and color. So some of these tomatoes only stay green. They never turn yellow or orange. Actually, none of them are red when they ripen. They're only ever orange or yellow or green. But the purpose of our project is using this computer vision tool that we measured to be able to phenotype a lot of these fruit all at once without having to take measurements by hand. So we don't want to have to, weigh each fruit individually or use a caliper or a ruler to measure each fruit individually. So instead, we developed this computer vision pipeline designed to be used on our wild tomatoes as well as all of the wild and cultivated tomatoes that we're growing in the field. But basically, what we do in our pipeline is we have this photo box here. It is equipped with a scale as well as a color checker, and we put all of the fruit in the photo box. We take just one photo, and then from that photo, we input it into our computer vision pipeline, and these are the outputs that we get. So we are able to first color correct the photo. We are able to scale it to the actual size of the tomatoes, and then we segment out each fruit individually, and we can measure the size, shape, and color of the fruit. So these plots over here are just verifying that our tool works as intended. So for example, this plot is showing the diameter measurements that we got from the photos using computer vision versus all of the hand measurements that we took for verification. And you can see that there's such a strong correlation that the computer vision tool can measure the traits of these fruit as accurately or more accurately than we can with hand measurements. So we also color correct. These two plots are just showing that we can color correct the photos before we process them to be more accurate color-wise, and then we can analyze all the color of the fruit. But what we're basically looking to do is look at how the traits of these fruit vary across the phylogeny. So these are the Galápagos Islands where all of the different samples that we're looking at came from. And we can look based on the tree to see, okay, well, it looks like these fruit from this purple island, Isabella, are tending to be more green. They don't turn yellow or orange while they ripen, but the ones from this Fernandina island do turn orange. We're looking at variation in size and shape, and eventually we're aiming to apply this pipeline to the almost 200 different varieties that we have growing in the field, and that's cultivated tomatoes as well as the wild tomatoes.
Wow, that's a lot of cool and interesting science that you've been doing this summer. So now, does this phylogeny and the colors associated with it, does that match what you were expecting?
That's a good question. So when we're looking at the phylogeny, it is expected, of course, to see that the samples that we take from the same island would probably be more closely related. And so therefore they would have, similar color, similar shape and size. But because these are wild tomatoes, there's a lot of variation, for example, in the environment of the islands. And so there is a lot of variation that we maybe wouldn't expect to see, or we're finding out maybe that some samples that were taken from closer together are more closely related to, distant samples. It's just interesting to look across the phylogeny.
Sorry. That's so exciting. I'm going to come back and ask you questions later because I have more, but for the sake of time, I think we have to move to the next one. Thank you so much. And congratulations on all the great research you've been doing.